Even very early embryos contain many different signaling molecules, which can make it challenging to discern the complete function of any individual embryonic signal. By isolating cells from endogenous signaling centers, these explants enable researchers to examine the role of a given signaling molecule in relative isolation.
The main advantage of this technique is that we are able to recapitulate embryonic timing, cell movements, and gene expression patterns within a simplified ex vivo system, without the time or effort required to maintain stem cells in culture. Begin by cutting the explants from the uninjected embryos first. If the explants extend in culture, then the cut was made too close to the yolk.
Begin by filling a pulled glass capillary needle with RNA. Place the filled needle into a micromanipulator and break the tip of the needle with the forceps. Calibrate injection volume using a stage micrometer with a drop of mineral oil, adjusting injection time and pressure on the pneumatic injector to achieve a bolus of the desired size. Keep the RNA needle tip submerged in the oil until ready to inject. Load the embryos into the injection plate using a Pasteur pipette and pipette pump, and then use a gloved finger to press the eggs into the troughs gently. Inject 10 picograms NDR2 RNA into the yolk of single-cell embryos until the desired number of embryos is reached or until embryos begin to divide. Use a gentle stream of egg water from a squeeze bottle to wash the embryos out of the injection plate into a labeled petri dish. Place the embryos into the 28.5 degrees Celsius incubator until they reach the 128 cell stage. Remove unfertilized eggs and